Good afternoon. Thank you for joining our American Academy of Periodontology webinar on COVID-19, separating fact from fiction, really looking at the scientific principles of COVID-19. So my name is Will Giannobile. I'm the chair of the Department of Periodontics and Oral Medicine at the University of Michigan School of Dentistry, and I also serve as the co-chair for the AAP's Future of Science Strategy Task Force. I'm joined today by Dr. Purnima Kumar, and Dr. Kumar is a professor of periodontology at The Ohio State University, and she's an expert in microbial pathogenesis and really linking together the role of the host defense with the oral microbiome. And so her work has been continuously funded by the National Institutes of Health for many years, really focused on many principles on microbiology. And so as just a bit of a background for today's webinar, it's going to last for about the presentation, 30 to 40 minutes, after which time we will have time for question and answer, and I will do my best through the chat feature to answer your questions. And so as a participant, our AAP members, as well as our guests who are joining us today, you will receive 1 CE credit, and then that credit will be applied to you over the next couple of days via email. So as we think about the novel coronavirus and its implications, back on March the 11th, the World Health Organization recognized COVID-19 as a global pandemic. And so during this time, according to today, the World Health Organization has identified over 800,000 documented cases, over 40,000 deaths associated with COVID-19 infection. And so here in the United States, there are many implications of COVID-19 with well over 160,000 cases and nearly 3,000 deaths. So as a part of this webinar, our goal as a part of the AAP is to serve to help educate our members and guests on the oral health implications for the better healthcare delivery of our patients. And so with that, without further ado, I would like to again introduce Dr. Purnima Kumar, who will really take us through all the biological aspects of COVID-19 and the implications to periodontal practice. So Dr. Kumar.
Thank you, Will. Hello, everyone. I hope you are well and staying safe. And you know what they say, right? When times are dark and everything is wrong, what do? We turn to science. And so today, even though there is very little science that has come out of this very new disease, we're going to see what we can learn from science that will help us separate all of these stories that are coming out in the media from what is really the fact of the disease. Something that began in January as 555 cases in a little isolated province in China grew in a month to 50,000 or more cases, came to the United States in March, and as of March 29th, there are 770,000 cases worldwide. United States, unfortunately, has the dubious distinction of having the most number of cases tested, and we have very little knowledge of this disease. There are so many questions that we can ask, and we do keep asking. And so just in the interest of keeping everything under 1 hour, I figured that we would look at this in 4 broad umbrellas. Let's look at the virus and its virulence, what we know about the biology of this virus. Let's look at the science of modeling disease contagion, this disease model that is keeping us here, that is keeping you in your home and me here and us talking through a computer. Let's look at the science behind aerosol contamination, because this is what has kept all of us out of our offices doing what we do best, which is making every patient's life better. And let's look at what the future holds for the vaccines and the drugs that have come out. Let's start with the virus. You hear so many names. We hear COVID, Corona, COVID, nCoV, all of these things. The disease is COVID-19. It stands for Coronavirus Disease 2019. It is caused by the severe acute respiratory syndrome coronavirus 2, SARS-CoV-2. This is the 7th coronavirus, and like all its 6 previous versions, it is zoonotic, which means it transmitted from animals to the human host. SARS-CoV, which caused the 2003 epidemic, MERS, the Middle East COVID, which caused 55 deaths in Saudi Arabia, and this new COVID, SARS-CoV-2, which is also known as a novel coronavirus, so nCoV, you see that designation in some things, nCoV and CoV-2 are the same. These are the ones that have caused severe disease. The previous one, the Hong Kong virus, the Nile virus, all of these others are known to have caused more mild versions of this disease. So when we look at the virus, excuse me, I'm just going to minimize my screen here so I can look at my screen better. All right, when we look at the virus is basically a single-stranded positive sense RNA virus. What does that mean? It means that the genetic material for this virus is enclosed in one single strand of RNA. And importantly, this RNA is a positive sense RNA. What that means is it goes from the 5' end to the 3' end, and because it does that, it resembles messenger RNA. Why is that important? Because it hijacks, because it looks like messenger RNA, it hijacks the host protein factory very, very easily, which is the ribosome, right? So this mRNA, the viral RNA, which resembles mRNA, hijacks the ribosomes to create viral proteins rather than host proteins. And because it is RNA rather than DNA, there are very few proofreading mechanisms. DNA has proofreading mechanisms when it's being assembled, when it's being transferred from parent to child, and therefore it doesn't mutate very easily. RNA doesn't have as many proofreading mechanisms, and because of that, mutations happen very easily. I don't know how big your screens are, but this basically is a genome map of the spike protein, and I'll talk to you about that in a second. And what you're looking at, these boxes that have been boxed in, these are the areas where the genes where there are mutations, and single nucleotide mutations have been changed here. Here you're looking at the human coronavirus. This is the bat. This is the pangolin, which is said to be the intermediate host for COVID-2. And all of these different viruses. So you're looking at how these mutations happen. These mutations happen very easily. And typically we have noticed that for the COVID-2, the mutations are happening in the spike protein and the accessory protein. And you're going to hear a lot more about this spike protein. Mutations enable this virus to jump species. Mutations happen for a reason. They happen as any species evolves and adapts to its new surroundings. And as the virus moves from a wild lifestyle to an intermediate vector to a domesticated animal, camel in the Middle East, the cow, the swine, the fowl, all of these different things, and then finally ends up in the human host. Each of these mutations enable this new lifestyle, and that's what happens with this. So let's talk about the spike protein. This is a photomicrograph. What you're looking at here is a photomicrograph of the COVID-2. It looks like it's wearing a crown, and that is because of this spike protein. A spike protein is basically a trimeric, so it has 3 little domains. It's a trimeric protein like that. And, that's there. It's a trimeric protein. So as you look at it, you will see it here. You see that there is one subunit in dark gray. There's a little gap here, almost like a ravine. Do you see that little gap? There's a second subunit sitting here, the second gray. There is a third subunit sitting all the way in the back here in white. So this is a 3 subunit, looks like a tripod all joined into one. So it's that kind of a protein. It's a spike, right? It's that kind of a protein. These subunits, I'm going to talk about the first and second subunits more than I'm going to talk about. Subunit 1, S1, contains a receptor binding domain. You see this green thing here, this green protein is the one that is the binding domain for the receptor. So this is the green domain. And this subunit is very important for the cell, for the virus to enter the cell, to initially enter the cell. The second subunit 2 is sitting right here. It is important when the viral membrane starts fusing to the cell membrane, and therefore it helps the virus to continue penetrating into the cell. Now, the reason why these 2 subunits are very important is for the virus to become virulent, for it to become pathogenic, for it to enter the host cell, a very important thing has to happen. And that is these two subunits have to get cleaved. That little lightning bolt is supposed to be the cleavage of these two subunits. They have to get cleaved. And that cleavage happens through a human protein known as furin. This is a protease. It basically takes any protein and breaks it down. This is found in humans. And you will notice one thing as I put this up. This is the protein expression score in females, in males, and this is the RNA. And you can look at the different organ systems in which this protein is expressed. You will see it's highly expressed in the brain. Look at what you see here. Every dentist here must be smiling. Tons of it is present in the salivary gland. Tons of it is expressed in the colon, liver, kidney, testes, lymph nodes. So furin is present everywhere in the body. And I'll explain to you why I'm focusing on the salivary gland. You will see why I'm focusing on the salivary gland in just a minute over here. So furin is expressed, it's ubiquitous. It is expressed everywhere in the human body by multiple different organs. Now, this takes on a special significance if that organ is facing the outside world. For example, lung. It's highly expressed in lung. It's highly expressed in the oral cavity. It's highly expressed in the gastrointestinal tract. It's not somewhat, very minimally expressed in the eye. Yes, it is expressed in the brain and the kidney and the pancreas, but those are internal organs. They are not world-facing. They're not front-facing. And so that doesn't become as important to the science of viral disease, of COVID-2 infection, as everything else. One more thing needs to happen. After the S1 and S2 subunits have been split by furin, the S2 subunit binds to its ligand, which is the angiotensin converting enzyme 2, ACE2. This, again, is a human enzyme. And I'm going to show you this is beautiful work. It looks a little fuzzy on my screen, but this shows that ACE2 can be seen in lung alveoli. It is found in the dermal regions, capillaries, salivary glands of oral mucosa. In the skin, it is found on hair follicles. It is found in your sebaceous glands. It's found in capillaries. It's found in the basal layer of epithelium. It is found in the glomeruli of kidney. So now you have a virus that is present, that has shifted, gone from having an animal host to a human host. You have two proteins that are ubiquitously expressed. So let us try and put all of this together to understand the virulence of this virus. First, it is zoonotic. Why is that important to us? Because as it came from an animal host, over a period of evolution, over a period of million years, it has been evolutionarily protected against certain human defense mechanisms. For example, bats don't have interferon gamma. Bats don't have TNF alpha. So these are things that the virus would never have seen during its lifestyle in a bat, right? So there are certain evolutionary protective mechanisms that this virus can use, and the human has not yet ever been evolutionarily been exposed to this. So this is new to the human, but the virus comes with certain very built-in advantages. It co-opts to ubiquitous human proteins for colonization. What does this mean? It can utilize multiple ports of entry. It can come in through your mouth. It can colonize through the gut. It can colonize through the eyes. It can colonize through the skin, hair follicles. So it has multiple portals of entry, and it can colonize multiple different organs. Another thing, and I didn't spend much time talking about it, but one thing that has come out, and this is very recent work, and it's beautiful work, is that it has different stages of the disease of COVID-2. Different stages of COVID-2, there's different tissue tropism. So during the acquisition phase, they have recovered tons of viral particles. And I'll actually show you the evidence as we go down this talk. During the acquisition phase, tons of viral particles can be found in salivary glands. In the early symptomatic phase, it seems to be completely localized to the upper respiratory tract. So it is in the nasopharynx, the oropharyngeal region here. As the disease gets more established, it seems to descend and stay in the pulmonary alveoli, and that is why we're getting all of these symptoms of pneumonia and pulmonary effusion, and all of those other symptoms are being seen. One thing that we will spend a lot of time on, and I just put this up here, is that it seems to have multiple modes of transmission. All the modes of transmission are not completely elucidated. We don't know yet for sure, but it seems that it can spread through direct contact. It's a droplet infection. It stays on surfaces, so it can spread through fomite transmission. There's also evidence that it can be found in the feces, although there is no record that a fecal oral transmission route has yet been established. So we don't know that there is a fecal oral transmission, but the first COVID has been isolated from feces. So great. When can I get out of my house, right? That's I'm sure you're all asking that because I have been asking that. We all need to know when we can get out of our house. And to help explain that a little bit, I am going to talk about the science of modeling disease contagion, because this is what everyone in the media is talking about every time all of our leaders talk about the disease model, the disease model. Well, what is it? What is it? It's not simply someone looking into a crystal ball. It's actually a math. It's a mathematical model. And what goes into this mathematical model? What happens is this. There is this person, patient Z. We should call this patient Z. This patient transmits disease to one other individual. And there you have what is known as an outbreak. There everything begins, right? Everyone else in orange has no disease, which means they are at risk. They are susceptible. Now you go to lots more people having disease. So all of these people are infected, and there are still some orange people here. And at this stage, one more thing happens. That is, this first person who got disease, one of these people who are infected gets removed from this equation. They either die or they get better. But with Ebola, we see a lot of them that they died. With other diseases, they start getting better. So somehow they get removed from this equation. So mathematicians take these three scenarios and put every population into these giant buckets, 3 giant buckets: people who are susceptible, people who are infected, and people who are removed. So you have the SIR. This is the SIR, right? This everybody in the population is put into one of 3 buckets. The models that I'll talk to you about today used one more group because this is COVID-2. They used a group that is known as exposed. That is people who are infected but are not yet showing symptoms, right? So now that we're getting data on this group, they added one more group. So we went from SIR to SIRE, and this is the model that is being used. Now, what happens when you use this model? Another important concept that I'm going to introduce to you is this R naught. R naught, you hear this, R naught. Every time you turn the video, if you turn any YouTube on, any talk, everybody's talking about R naught. What is R naught? It is basic reproduction number. What it simply means is that if I am sick, how many additional people can I get sick in a population, assuming that nobody in the population has immunity, that this is a novel disease, it's the first time it's come in, nobody has seen this disease before, and therefore nobody in the population has immunity. How many people can I get infected? If my R naught is less than 1, I get the disease, I get better, or I die, and I'm done. If my R naught is equal to 1, I get disease, I transmit it to one person who transmits it to one more person. The problem only happens when R naught becomes greater than 1. As soon as R naught becomes greater than 1, you start having what are known as outbreaks of disease. So they can be localized, they can be endemic, they can be epidemic, and then they can become pandemic. So all of these things. As you go from R naught 1 to R naught 2.5, there's me in blue. I only infected 4 people, but look at the network of infectivity these 4 people created. And R naught of 4, if you look at R naught of 4, this went exponentially. There's no linear number here. It's not even an exponential number. It's some kind of a sine, cos, tan, whatever you want to add. There's some kind of a math that's going on that kind of builds to this. So that is what R naught tells you. To give you a perspective, R naught of measles, one of the most contagious diseases, is 23. The first SARS was 2 to 4, and COVID-19, they have estimated it to be about 2 to 3. R naught values are not gospel. They depend on many different things. The first thing is rate of contact. How many times do I go out of my house and shake hands with people? How many times do I see a patient? How many times do I do X, Y, and Z? It also depends on the rate of transmission per contact. Remember that Biogen meeting in Massachusetts where this one person got 74 people infected? That is the rate of transmission. Remember the case in Korea where they all went for choir practice and 55 of them went home with the disease? Those kinds of things, right? That is the rate of transmission per contact. And again, it looks great. It says, oh, one person can give it to 25 people. No, That number depends on a million other factors. It depends on the person who's transmitting the disease. It depends on the cohort that might be susceptible to the disease. It depends on whether they are in a closed room or whether they're playing football outside. It depends on what their natural immunity to the system is. It depends on many, many different things. The other important variable that an R0 depends on is symptomaticity. That is, what are the symptoms of this disease? Before, when the first time they calculated R0, they thought fever was the first symptom of the disease, and therefore people who did not have the fever were considered as safe. And so that is how R0. Now we are finding out that maybe it is anosmia, loss of smell. Maybe it is a GI, stomach flu. Maybe we don't even know. So we'll get to that in a second. So all of these keep changing. The fourth important factor is virus biology. And so R0 values have to be specifically computed for each specific infection. The virus, how long does it survive on an abiotic surface? So I sneezed and I walked away. How long is the virus alive? 10 minutes, 5 minutes? What kills it? Can I simply kill it with bleach? Do I need a complex, antimicrobial to wipe it off? What is a droplet contamination zone? If I sneeze, how far are these droplets carried? What is the mode of transmission? And most importantly, what is the window of infectivity? If I come into contact with a patient today, do I go home and am I immediately infected? Or do I have to wait a few days for my prodromal phase to get done and then for this infection to start again? So what is my window of infectivity? And because of that, our values keep changing. People become immune. As the disease progresses, people start getting immunity to the disease. And that is what was happening. The whole UK story depended on that. People in Britain didn't take much of an action in the beginning. When my nephew lives in Britain and he was going to school when we had shut down here. And the reason was they thought herd immunity will kick in and everyone will get better. They thought that people in England had already gotten some level of disease, that half the population had been exposed to the disease and herd immunity had already kicked in. So different things, people can get immune. People's behavior change. We are all on our best behavior. We are isolating. We are shelter in place. We're doing wonderful things. What happens when they say they get sound the all clear and we all get out again? As we go out, are we going to cause a resurgence of this disease? We don't know. But the most important reason why an R value changes is because an R0 is calculated in bug time. It's calculated in the virus time. Dr. Fauci made a fantastic statement when he said, the virus decides when this quarantine ends. We don't decide. People don't decide. That's exactly it. For this infection, the period of infectivity we thought it was 7 to 9 days. So it made sense if an R0 was calculated every 7 to 9 days. Now it's gone to 19 days. So maybe we should calculate R0 in 19 days and not look at it on a daily basis. So if you look at the R0 daily, it becomes inaccurate. Like every periodontist in this group will recognize this, right? You can do the PSR once as your patient shows up in your chair for the first time as a screening tool. R0 can be calculated once. It makes certain fundamental assumptions that nobody has immunity to the infection. As things change, as behaviors change, R0 values will change. Two models, we've always heard about this. Everybody, two people have been vilified for this, the sky is falling kind of a doomsday prophecy model. The Lipsitch model that came out suggested that 660,000 Americans will get seriously ill or die, and that we might have a second surge in autumn 2020. The Ferguson model said something similar, that there will be 200,000 deaths in the United Kingdom, and that once social distancing rules would be relaxed, there would be a recurring surge of the same disease, right? The Ferguson model was really, people talked about it a lot in the media because they said that, the Ferguson model recalculated to go from 200,000 deaths to 20,000 deaths. They did not really do that. If you look at the data very closely, what they did, what every good model should do. They took an initial set of assumptions and gave you a model before the country went into quarantine. The assumptions that made the initial models were that the entire population is susceptible, that the R0 is really low, that this disease spreads, but people are getting mild symptoms, nobody's dying off like crazy. It's not the Black Death, right? And then population density estimates. Remember how I said the rate of contact, how many people can I infect in a day? That depends on how many people live closely in all different things, right? So population density estimates. The first density estimates came to us from Wuhan. They came to us from Italy. And we had the best floating laboratory in the world. I'm not being facetious, but there can be no other study that was going to have data like this. The Diamond Princess cruise, 1,300 people quarantined in place, in shelter, isolated from everything else in the world. That became a floating lab. And so that allowed us to, estimate population density. And that's what we started. And when this first model was made, there was no social distancing. And what we thought was, like most viral infections, the infectivity period will be about 7 to 9 days. And one of the early assumptions made was that early vaccines are not going to work against this disease, which is why the second surge and the surge in fall and all of those stories keep coming up. Now, we know that it is not R naught, but RE, which is R of exposure. There are people who have been exposed to it. We know now that the symptom complex this disease presents with is very complex. You can have loss of smell, you can have gastrointestinal disturbances, you can have a mild cough. There are people reporting nothing more than sniffles. It doesn't have to be fever, right? Temperature is no longer a pathognomonic sign of infectivity, of symptomatology for this disease. We're also finding out that the infectivity period can be anything as long as 19 days. Most importantly, we're finding that asymptomatic people can transmit disease, that you can be completely symptom-free and you can still be a carrier for disease. We're finding that vaccines will not be available for 12 to 18 months, which is allowing us to recalculate all of our surge models. And most importantly, that allowed people to change their models is that certain groups are more vulnerable to others. We're finding that, for example, males are being, affected more than females. Smokers are being affected more than, non-smokers. Older individuals are being more affected. Children are not being affected. So as soon as you start taking the entire population and start breaking it into little subgroups, you realize that, the population is not the population. That small subgroup is the population. So of course, your R naught has to change because now your number went from 6 billion people to maybe 300,000 people. So those things change. And that's how this changed. Now, to be accurate, why do we care about this model? Because that date, remember, they said March 15th, they said April 30th. Somebody just yesterday said June 1. It seems to be going to infinity. This self-isolation seems to be going on. And so we're desperately looking to this model to be accurate. We are hoping for that model to be accurate. What is going to be needed is data on how many people actually social distance, which means that people are going to be looking at your phones. There are going to be companies, there's going to be people who are looking at your phone tracking to find out where you went on what day and how long you traveled and how long you quarantined when you said you did. Another thing we don't know is data on children. Our children, how much do children transmit? What is going on? Who are the asymptomatics? Are they truly the asymptomatics? Can people with mild symptoms be calculated as asymptomatics? And so to identify truly who the asymptomatics are, we need serological testing. We need serological testing of this S group, the susceptible group. Remember that S group? We need serological tests. People are buying serological tests as they come into market. So yesterday there was a breaking news of 135 kits that were sold to Georgia. Each state is buying this test as it comes out, and that is going to improve accuracy. And in the absence of vaccines, what we're doing is we're taking FDA-approved drugs, non-approved drugs, kind of word-of-mouth therapy, anecdotal evidence, and we are treating these people. We need to know, as people get treated, what do they get? They get immune to the disease. So as we get data on efficacy of pharmacotherapy, this model will get more accurate, and we will know sooner when we're going to get out of our homes and get our lives back. In the meantime, only 2 non-pharmacotherapy approaches are available to us. 1 is limiting exposure, which means stay where you are. 2 is limiting contagion, which takes me to what we are all thinking. Did I really have to close my practice and sit here listening to Purnima talk about this? I'm going to divide this. This is a giant topic, right? So I'm going to divide this up into 3 smaller bits. We're going to talk about SARS-CoV in saliva. We're going to talk about the modes of transmission, and we're going to talk about the survival of the virus on abiotic surfaces. COVID-2 can be found in saliva, human saliva, in symptomatic patients and in asymptomatic patients. It is found. The source seems to be the upper respiratory tract from, coughing and sneezing. Minor and major salivary glands. These are two animal studies, but remember where you found the furin and where you found ACE2? Tons of them are found in the salivary glands. So it seems like the major and minor salivary glands are big reservoirs. And every periodontist in this group is going to groan. Every hygienist in this group is going to groan. There is anecdotal evidence, not direct evidence. There's anecdotal evidence that the gingival crevicular fluid might be a source of COVID-2. Modes of transmission. A virus can be transmitted in many different ways. Droplet infection. You all know what a droplet infection is. It is carried in the saliva, mucus, or sputum. What that means is that someone has to be within 6 feet of a person for a droplet infection to work. The other way is an airborne infection. Airborne means viruses travel on dust particles. They are not just on droplets or mucus, which is heavy, gunky things that fall, that plop where they come out. These travel on dust particles. They're light, they're micron, submicron, even supermicron levels, and so they can travel more than 1 meter. What that means is the patient doesn't have to be in contact, or the person doesn't have to be in contact with the patient. It can travel through many different sources. It can travel through many different resources. And more importantly, it can be transmitted over extended periods of time, Let's talk about airborne transmission, because this is what is keeping us here today out of our offices. What is the science behind airborne transmission? The WHO report, 75,000 COVID-19 cases analyzed, airborne transmission was not reported. The Ong study from Singapore, it was a field study of 3 patients, 2 weeks follow-up, 26 sites were sampled, the air samples were negative, exhaust outlets were positive, which means that, this can be transmitted, the droplets can settle somewhere, that can happen. The Yuanlu study was a Wuhan field study of 2 hospitals. One of the hospitals saw 200 patients, few or no airborne viral readouts. The readouts decreased when the hospitals became less crowded. Aerosol gets deposited up to 3 meters from the patient and can accumulate over 7 days. This is one study that gets talked about a lot. This came in the New England Journal of Medicine on March 17th, last 2 weeks ago maybe. And this is an in vitro simulation. They took a simulator, an aerosol simulator, and they sprayed an environment, a contained environment with the virus. And they found that the virus is viable in aerosol for 3 hours. It gets transmitted, the virus that is transmitted is viable on many abiotic surfaces for days. But the questions that this does not answer is, 1, the doses that they used for this aerosol are not considered physiologic. And as if you read the WHO report on this study, the WHO says that aerosol generation is not compatible with human coughing or sneezing topography, which means the way they generate the aerosol is not how humans cough or sneeze. And so that is that study. So we need to think about what that study tells us as we decide where we're going from this, right? The third mode of transmission is direct contact. Touch you, touch me, touch my mouth, touch my face, touch my face, touch my mouth, those kinds of contacts. And an indirect contact is the aerosol spreads, touches a surface, you touch the surface, you put your hand, that's called a fomite transfer. So you can have a fomite transfer or you can have a direct transmission. This is that NEJM study that I just talked to you about. This is that in vitro study, and here's the graph. On the top, the brown bar here is the COVID-2. The blue lines here are the COVID-1. On the x-axis, you're looking at time in hours. And on the y-axis, you're looking at virus titer, right, per liter of air. So this is 3 hours. Both COVID-1 and COVID-2 can persist in aerosol over 3 hours. And then when that comes on copper, it's gone within 20 hours. Cardboard is longer for about 60 hours. Stainless steel and plastic are 80 hours. What's that? 72 hours is 3 days. So over 3 days, right? So they can persist on stainless steel and plastic viably for over 3 days. However, there are other studies that show that these viruses can be inactivated. And this is what they have tested: 62 to 71% ethanol for 1 minute, 0.5% hydrogen peroxide for 1 minute, 0.1% sodium hypochlorite for 1 minute can inactivate these viruses. And so, to summarize all of this, aerosol transmission is not a currently accepted mode of spread, but viruses can exist in aerosol form. Aerosols can deposit on abiotic surfaces, and from there they can be transmitted to other people, unsuspecting, people, and that is feasible, it is plausible. Procedures that generate aerosol extend the contamination zone by 3 meters and extend contamination time by 4 days. Importantly, asymptomatic and mildly symptomatic individuals have similar viral loads, so that patients sitting in a chair that, undergoing an aerosol generating procedure might generate a similar load to one who actually has tested positive for COVID. Only serology can provide this window, can provide us an accurate window, which means your R0 can change, which means if we conduct aerosol generating procedures and that spreads the infectivity and the zone of contamination, our shelter in place times can increase significantly. Dentistry, what are we going to do? We're going to have a wonderful talk by Todd. We're going to have another wonderful talk by Mia. So I'm not going to spend a lot of time talking about this. You know what they say, right? She who looks at crystal ball is destined to eat shards. So this is my prediction. The virus has already jumped species. It is here to stay. All humans are going to have this virus. There's going to be a potential requirement for more stringent infection controls. I see that coming down the pipeline, right? Maybe we're going to implement more pre-screening. Maybe we'll make questionnaires that are living documents, and as we come to a new disease, we'll add more questions pertinent to the disease. Maybe we will look for seropositivities, serological typing of patients for screening. Maybe we will start, and this paper that came out, the Peng et al. Paper that came out in the Journal of Oral Science on March 3rd, actually advocates all of these protections for dental professionals in routine situations, right? And so what we really need, what we are really missing, and the reason you and I are stuck here talking over a phone is because we don't have research. We have no research that tells us that dentally generated aerosols, that orally generated aerosols, that aerosols generated from dental procedures transmit COVID too. We also have no evidence to show that they don't. And so absence of evidence is not evidence of absence. We have no research, zero primary research on dental aerosols, zero research on carrier states among dental personnel. How many dentists do we know? Is there a database of dentists who are tested seropositive? There's a database of medical personnel, and there's a beautiful study from the Dutch group that has come out. We have nothing like that here, comparable, right? What is the patient Z spread cloud? Patient Z got treated, walked out of your office. What was their spread cloud? If and something simple as this, we talk about abiotic surfaces and this big scare. What is the role of your toothbrush in reinfection? You got the disease. Did you change your toothbrush? Did you do that? We don't have any questions. We don't have any answers to those questions. So I'm going to touch very, very briefly on chemotherapeutics because I know we're seriously running out of time here. There are lots of different things. Convalescent serum is one that's being considered. This is people who have had the virus and have recovered from it. So they contain tons of antibodies. So convalescent serum is something that is being seriously considered. There are 332 proteins that are connected to COVID-2 that are being tested. 69 drugs can target these 332 proteins. 27 of them are already FDA-approved drugs. Breaking news, there's going to be no vaccine in 2020. The fact is that this has a nice lipid layer, and so soap breaks up lipid bilayers, and so hand washing is key. If this virus spreads by contact, soap can break up the viral outer membrane, and therefore the fact is that hand washing is key. There is a new thing that came out that said, please stop taking your ACE inhibitors because that causes the disease to spread. that is fiction. Please continue taking your medication. The WHO, the American Heart Association, the recommendation is that you need to continue taking your medication. So this is what happens. As soon as the virus uses your ACE2 to gain entry into the cell, it down-regulates ACE2. Now, fundamentally what happens is angiotensin 1, ACE, converts angiotensin 1 to angiotensin 2, which then goes down and causes vasoconstriction, increases vascular permeability, and this is how this happens. Your ACE inhibitors, those medications that you take, inhibit ACE here, so prevent conversion of angiotensin 1 to angiotensin 2, right? If you stop taking this medication, ACE2 does one more thing. Now, let's step back here. ACE gets downregulated because there's a viral infection. ACE2 does something very important. It takes angiotensin 1 and breaks it down to angiotensin 1,9. It takes angiotensin 2 and breaks it down into angiotensin 1,7. So it prevents this cycle of injury from happening. Now, when you have a viral infection and your ACE2 is downregulated, this byproduct breakdown does not happen, which means your body is going to depend on that ACE inhibitor that you take for ACE to stop continuing this process. So there's a lot of evidence that shows that angiotensin receptor blockers, ACE inhibitors, all of them play an important role in keeping the lung injury down following a coronavirus infection. Don't take NSAIDs. That again seems to be, that's why I've put this in a very faint thing. It appears not to have a lot of basis in science. Prostaglandins work in many different ways. Some of them inhibit, some of them promote, some of them attenuate lymphocytic response. And so the current wisdom is that people taking NSAIDs for other reasons should not stop. Chloroquine, this is control. This is hydrochloroquine. This is the PCR positive samples over day 6. Chloroquine, hydrochloroquine with azithromycin seems to be working fantastically. It does seem to be working. And there's a biological mechanism. Chloroquine actually alters the surface of the cell membrane, so it prevents the virus from fusing to the host membrane. It inhibits nucleic acid replication. And remember, this is a viral RNA. This is an RNA virus. It's a plus RNA virus, right? So it inhibits nucleic acid replication. It does many different things. So it is plausible, biologically plausible that chloroquine might work. Same thing. Remdesivir is another one that is. What remdesivir is, it's an adenosine analog, which means that, remember those 4 nucleotides, A, T, C, G, it can bind to the nascent mRNA of a newly forming virus. And because of that, that virus stops. That virus replication stops. And therefore, viral replication is stopped. Remdesivir has been tested for Ebola and other coronaviruses. So it is plausible that remdesivir can work. 6 different vaccines are now in production. This is against spike protein. 2 of them are in phase 1 trial. This is out of Oxford. This is in a phase 2 trial. These 3 are still in the preclinical testing phase. So to summarize, the virulence is highly contagious. It spreads through both direct contact and through droplet infection. The reason behind, we are staying here is that all disease models, no matter how you slice and dice them, consistently demonstrate the impact of lockdown and distancing. There is no evidence that dental aerosols spread virus, but there is no evidence that it doesn't either. And serology and vaccines are critical to early identification. Until then, treat everyone as if contaminated. And in the meanwhile, I wish you the best of health and good cheer. Stay safe, be well. And if there's time for questions, Will, I will turn this over to you, and thank you for your attention today.
Okay. Well, thank you, Dr. Kumar. This was really a tremendous presentation. It was truly a tour de force in terms of covering all of the aspects of the basic biology and many of the implications that we, with the best evidence, you've got, data that you're presenting, they're only a few days old. So we really appreciate this presentation. So we've had a lot of excellent questions that have been coming forward. One of the questions that's been repeated in a couple of different ways is you talk about when a patient gets infected, what actually happens, and what type of immunity. I know that there's some studies in non-human primates that have suggested that there is a modest antibody response, and many people speculate that it's possible that you would develop some modest response, and then if you got reinfected, the symptoms would be less.
Right.
What do you think? What is our best evidence right now on this reinfection?
Right. And as you said, Will, the best evidence is right now only from animal studies. But there's a huge push for convalescent serum, and there's evidence coming out of Italy that suggests that convalescent serum, people who are who have recovered from the disease, there's obviously some factor, they don't know what the factor is, but there is some factor that convalescent serum. And so just like when the people were doing fecal transplants, they don't know what part of the fecal transplant actually works. They are pushing the whole thing in. Similarly, right now, we are in war mode. I hate to use that word, but we are really in battle mode. And for that, they are using convalescent serum. People are like, act now and ask questions later. So yes, there seems to be a modest antibody response from animal studies. The sister virus CoV-1, there was a very nice, robust antibody response, which showed that reinfection was a much simpler, much more easier to treat disease. So they're hoping that this would be similar to that. There's really no evidence. At this point, the evidence is very slender, and to say anything would be to falsify data. So we just have to wait for things to play out and sit back and keep ourselves away from other people.
Okay. So along those lines, Purnima, as you talk about, this truly as a pandemic, a large proportion of the population is likely to get affected with this virus. And so the whole concept of herd immunity, do you want to explain this to individuals? Because people are asking about, we're looking at these spikes of infection in many different communities, and people are trying to quote unquote flatten the curve and looking at ways, the self-isolation procedures are helping, but after a period of time, there's going to be a good number of people infected. I know this is somewhat related to the last point, but do you think that is something of consideration, like looking a year from now, if we don't have a vaccine, how are we going to be functioning day to day?
Right. So the best thing about this disease is that it's a viral infection, right? So just as it spreads virally, it also and you know what? So one of my friends actually made a very, very facetious remark the other day. She said, since we're listening to the CDC now, let's all of us get vaccinated, right? Let's get vaccinated for our other viral diseases. Let's and I'm not getting on the anti-vaxxer soapbox here, but that's the whole concept, right? When people develop, when a whole group of people develop immunity to a viral disease, they act as a barrier. It's almost like a penguin huddle, right? You start developing these boundaries, you start developing walls inside which the most susceptible people can shelter. And so people who cannot get immune, who for other reasons are somehow immunocompromised, who are susceptible, who cannot get vaccinated, if you don't carry the disease, or if you're not, if you're resistant to the disease, you stop being a transmitter, you stop being a vector, and you may be healthy. That's not the issue. You can be healthy. I mean, I can't remember the last time I got a cough or a cold, right? Some of us are naturally resistant to this, but that doesn't mean I am not a carrier. And by carrying that disease and transmitting it to someone who cannot get vaccinated, I am actually being that typhoid Mary, right? And that's what herd immunity does. It develops a wall, a communal, a people wall around people who cannot, for many different reasons, develop that immunity for themselves or acquire that immunity through other reasons.
Okay, no, that's very helpful. Now I'm going to switch topics a little bit. We've gotten a lot of questions that, within dental practices, a large number of them are shut down and really focused on emergency care. And we've seen from the studies in Wuhan and others using different types of PPE. So do you want to give kind of a summary on, you highlighted that study from New England Journal on aerosols and how they can, the virus can last on surfaces for periods of time. But do you want to talk just briefly on best practices right now? We know this is going to change next week, but right now on oral rinses, and then the other one is in disinfection of operatories.
Right. So the two things that we can really hang our hats on right now is, one, we are masters of decontamination. I mean, we are dentists. We live in aerosol-rich zones. And I want to know how many of our patients walk out and fall dead from infections. They don't. So we are masters. We know how this is done. We got this. We got this. What we need to be a little, so we have the tools. We have the tools. What we need to do is, implement these tools, weaponize these tools, right? The one thing we have to do, we have to be very cognizant of, is that this virus can live on abiotic surfaces for up to 4 days, which means this virus can live in an aerosol for up to 3 hours. So if your patient walks in, you might not get infected. I mean, the big question everyone is, I have an N95 mask, can I go practice tomorrow? Well, the N95 mask protects you. It protects you, the practitioner. There's your assistant suctioning on that side. And by the way, are you wearing a hat to cover your hair? Remember, your hair follicles, they contain a ton of furans. And they contain ACE2 receptors. So you know what? They might settle down there. They might settle down on different body parts. Are your shoes permeable? Is your footwear impermeable? We don't talk about footwear. We don't consider those things, right? Are the gowns you wear, what is the permeability level of the gowns that we are currently using in our practices? We don't know. When will you schedule your next patient? Are you going to wait 3 hours between each patient? Yes, negative airflow. Everybody says, I'm going to install negative airflow, vents in my clinic, airflow, and that's going to be fine. What negative airflow does is it prevents the infection from going from inside your office to the patient waiting in the hallway. It's not a vacuum. It doesn't suction out the air and replace it with new air. It just doesn't do that. That's all negative airflow devices do. So they don't cleanse your office for you. They don't. So we need to start thinking about these kinds of best practices. What is it? And using more common sense and coming together as a group to start putting together best practices. I mean, this definitely has changed how we practice dentistry. I'm sure there are going to be long-term consequences to what we do. We just cannot practice dentistry in a vacuum. We cannot practice dentistry in, infectious disease operating rooms. That's not dentistry. We are in a highly, contaminated environment. We do crazy surgeries in highly contaminated. I mean, I'm a microbiologist. I cut and I sew in an ecosystem that has as many bacteria as the entire gut. Nothing happens, but we have to be mindful of certain things and change how we practice. And how do we change the practice? I don't know that we know yet. Should we change those rules? Absolutely.
Okay, and then extending on into the oral cavity with the patients, what types of pre-treatment rinses? There's been a discussion on things ranging from chlorhexidine, sodium hypochlorite, hydrogen peroxide. What are some of your thoughts there?
So two things have emerged, right? The JDR study, the one that we recently published in JDR. They recommended that they found that chlorhexidine didn't seem to work. The dental practices from Wuhan seemed to show that chlorhexidine was not as effective. However, povidone iodine, 0.2%. Remember painting everyone's, I'm old enough to remember painting everyone's teeth brown and their gums brown back in the day. Povidone iodine seems to work, and 1% sodium hydrogen peroxide for 1 minute. So pre-rinsing with each of these two seems to be one practice that they have found to be efficacious. So it's a quick and easy thing to adopt as we figure things out for more long term. So that does seem to be something that, there is evidence to show it, there's data to support it. We might as well use science to back our practice right now.
So what do you think? We've got many dental hygienists listening to this webinar as well, and we talk about in contemporary dental hygiene practice, talk about an aerosol-rich environment. What would you speculate as we move forward, coming out of emergency-only care and how the practice might change in the interim period on the use of aerosol-generating devices?
You know, I hate to hazard a guess. Thankfully, I'm not a policymaker. I'd hate to be in the shoes of a policymaker right now. Yes, aerosol generating devices have been with us for as long as, you know, they were made for dental purposes. They wouldn't have an existence without, you know, us using, but I think we're going to be using some things like, you know, positive retraction, anti-retraction handpieces. I think we are going to come up with possibly new methods of making sure we're infection free. Remember back in the day when the dental water lines, dental biofilms issue came up and people changed how we took care of our water lines. Those kinds of practices, I think this is a good wake-up call to us, and it will serve to make us stronger and help us, you know, do better things for our patients, I think.
Okay. All right. Well, you know, we have many other questions, and I know it's unfortunately we're running out of time. But this has been, you know, truly outstanding to hear your perspectives, and I think to have such a seminar that's so in depth, and you know, this virus and the knowledge about it has only been around for a couple of months, and so I really think of this as a world-class seminar that you've been able to provide to our membership. So we're very grateful. I would like to give you a chance for, you know, one summation that you might have for our listeners and the people who are watching today as we move forward.
You know, I don't know. I think we're all in this together, right? We're all in this together. We have, well, you and I know this, we have students not knowing if they will graduate. We have residents not knowing, you know, people with jobs waiting on the other end, loans to pay. We have students who graduated, our last year's graduates just started their practice. I went to someone's open house literally 2 days before quarantine hit. We're all in this. We're all in this together, and there is no evidence. There is no evidence. The best thing we can do for ourselves is start generating the evidence. Dentistry has to be more proactive about generating that evidence, you know, or somebody will make our decisions for us, and we cannot afford to have someone else make the call on our behalf. It just happened. It just happened. We can't let this happen again.
Okay, well, very well said. What I would like to do is draw the rest of the listeners and the people watching today to 2 more COVID-19 webinars that the AAP will be hosting. The next one is on Friday. It's on Wednesday, April the 8th. It'll be held at 12 p.m. Central Time. And the title of this webinar is There Something in the Air? Infection, Prevention, and Control in the Era of COVID-19. So, you know, please tune in to that webinar if you want to have more questions on that. Purnima, you will be moderating that session, and Dr. Mia Geisinger will be doing the presentation. And then today, the Academy just added another webinar that will be held on Friday, April the 10th at 1 p.m. Central. It'll be held by Drs. Todd Shire and George Mandelaris. It'll be moderated by Chris Richardson. And this will be entitled From Perio During the COVID-19 Era, Working Within the Trenches, and How Will We Survive? And so I think that this particular webinar that will be held on April the 10th is going to talk about, you know, we have so many economic consequences on getting our practices back online. And so George Mandelaris and Todd Shire will try to give us some more information on that. So as I sum up in the last few minutes, today's webinar, for those of you who might have joined a little bit later, this will be made available on the e-learning center for members access within about a week from now. And again, we would like to thank you for joining us. Again, I thank Purnima for your outstanding presentation, taking your time for us today and educating us. And I would like to thank all of you listeners who've joined us today to learn more about COVID-19 and how we can get through this crisis. And we wish you all well, and please be safe. Take care.
Thank you, everyone.